Hello everyone, good evening to you and welcome to my last session, which this evening is on the nervous system. So, okay, what you know by now, I'm sure, is the format for the webinar. So just watch out for the pauses, because if you're playing this back afterwards, then actually you'll know that I'm just leaving you time there for you to answer, to stop the presentation and give yourself time to answer thoroughly, because in 40 minutes we haven't got time to sort of do that completely. So what am I going to focus on in today's session then? Well, I've picked 3 areas of the nervous system and I've tried to do as much as I can in terms of pulling things together and revising. I will inevitably miss little bits out, but I've tried to give you the feel for as much of it as possible. So there's going to be a focus on reflex arc and looking at structures of the neurons and nerve nets. We'll look at the sort of the vertebrate neuron structure compared to something like a nerve net in Hydra, which is a Cnidarian. And then we'll look at the nerve itself and how it's transmitted. So we'll look at the features there, the all or nothing response, etc., resting potential, you know, the main phases of the action potential graph that, you know, I'm sure you'd be used to seeing. And then finally, we're going to do some work on how it is that certain chemicals, certain drugs will affect the actions of the synapse and how that works. And of course, as usual, what we'll do is we'll round all of that off with a focus on past paper questions. And then I'll just say a little bit more today about the dice games that, you know, I'll leave you with. So here we go then. So just some recap key terms really, because I'm going to talk about the sort of neural pathways. So we've got some keywords there: stimuli, receptors, reflex arc, effectors and response. And what I'm going to do, I'm just going to pause, because what I'd like you to do is to think about which words are linked with which definitions there. Okay, so slight pause before I reveal. And just to reveal the connections, so stimuli. So yes, those are changes that can be detected by the body. It's so important, isn't it, that any change is detected. That's of utmost importance for survival. Receptors, those are cells that will detect the changes, and we'll talk about you know, those in various areas of the body. The reflex arc is that pathway where we know that we have 3 types of neuron cells connected together in a sort of pathway, and actually that is what leads to a very rapid and automatic response. Effectors, those are muscles or glands that will carry out the response for the body, and the response obviously then follows how the body reacts to a stimulus. So some key terms then to start with. So we know it's connected together like this. If there's a stimulus, a change in the environment, then that will be detected by a receptor, and that will then, if the stimulus is strong enough, that will generate a potential. We call it a generator potential, and that will cause the instigation of a nerve impulse, which passes along a sensory neuron. That sensory neuron will actually pass into the central nervous system. And we know that's the brain or the spinal cord if we're talking about a vertebrate like ourselves. And the impulse will then pass through a relay neuron, sometimes called an association neuron or an interneuron. Again, it's biology, so why not just have a few words instead of just one word to describe something. And then the impulse will travel out of the central nervous system along a motor neuron to an effector. So if it's a muscle, it'll contract. If it's a gland, it'll secrete a substance possibly, and that will be your response. I will just say to start with, as far as the spelling of neuron is concerned, I always put a sort of an E on the end of it, but you will see it in various places either with or without an E, but don't worry about it, you know, but just want to explain what I'm doing. We've got, as part of our vertebrate nervous system then, a central nervous system, which I've already talked about, which is the brain and the spinal cord, and the peripheral, and that's all of the nerves which actually come out from your brain, say to your eyes, or then from the spinal cord to the tips of your fingers or the tips of your toes. So just remember, CNS and PNS, you know, they're connected, but they're different. You may also, as well, be required to label something like a section or a part of a section. You wouldn't be lucky enough to have a question where you had to label everything for 5 or maybe even 10 marks. That wouldn't happen, but there might be 1 or 2 parts that you have to label. So if you see a section like that, then you need to be able to identify the grey matter here in the middle in this H zone, central canal. We can't see it there, but it's in there. The white matter, and then to know the difference between the dorsal sides, which is sort of on the top here, the dorsal roots, and the ventral roots, which are on the bottom. Okay, so some basics there. So what I've done is I've just left this in now, and I'm going to pause in a little bit to just say to you, right, can you now say all of those words? Can you actually label all of those parts and then go back and check for yourself that you've got it right as part of your revision for a test? So I'll just pause. Okay, well done. And now I'm going to move on. So the reflex actions, as I've said, they're very, very rapid. They're protective. They're involuntary. We don't have to think about them. And you can see here, as I've said, the way different diagram to show how it is that actually they're connected. And you can see the same sort of thing is happening on the other side of this section through the spinal cord, but we've got the dorsal and the ventral nerves here. And again, we've got the sensory neuron going in towards the nervous system. Look at the position of the cell body, and we'll talk about the structure of that now. And then we've got the motor neuron coming out from the nervous system. Again, note the position of the cell body there and in the association neuron, because that is very often the subject of some questions as well. So what happens as part of a reflex arc, you can imagine, is a stimulus, a sort of a change. So that could be touching a hot plate, it could be putting your hand or your foot on a sharp object like a pin or something. And then if that stimulus is large enough, if it's great enough, what will happen is that we'll generate a potential, as I've said, which will set up a nerve impulse, which actually causes the nerve membrane to change its polarization. I'll talk about that in a little bit. The impulse will travel along the sensory neuron, and you can see the cell body of the sensory neuron is just outside of this main part of the central nervous system here in this dorsal root in a swelling called a ganglion. The nerve impulse goes into the central nervous system. It changes and slows slightly because it then has to move over what I might describe as a chemical gap, the synapse, before then the nerve impulse then being set up again in this relay neuron. Then what happens is it passes over another synapse, and then a nerve impulse is set up along a motor neuron, which comes out and connects, in this case, to a muscle to move away from the stimulus. But as I've said, it could be a gland equally. So that's sort of a revision then of the reflex arc. So again, what you'll need to be able to do is to make certain that you can label these parts of the reflex response. So what I'll do is I'll just pause now to give yourself enough opportunity to think about what A through to E are, and then I'll reveal it. Okay, so I'm just going to pause so that if you're playing this back later, then you can actually label these for yourself. Great, so what we'll do now is we'll sort of go back over it, because the A was the sensory neuron, the cell body was B, white matter was C, D was the relay neuron, or as I say, interneuron, association neuron. All of those are acceptable. And then the E is the motor neuron. So as I say, simple questions, simple bit of recall could generate you a couple of marks in a test or an exam. Absolutely essential that you've got those sorted. So another thing that I just wanted to tell you about as well, because it's in the specification, and it's something called the meninges. And the meninges are just 3 membranes, and you find them in the brain. They're actually, they're sort of the dura mater, the arachnoid, and the pia mater. Okay, so they're just membranes which actually sort of line these areas of the brain, but also down into the spinal cord as well. Okay. I'm not going to say any more than that. I'm going to move on fairly quickly because I want to talk about a motor neuron. And as we know, the motor neuron is the nerve cell which actually transmits the impulse out from the central nervous system to the effectors, to the muscles or the gland. And again, recognizable parts here, which we will talk about because they form quite a lot of answers to questions. And that is the myelin sheath, which is a fatty substance produced by Schwann cells. The nodes of Ranvier, named after the French scientist who discovered them, Louis-Antoine Ranvier, and so they are gaps in the myelin. And you'll also see dendrites, and because the nerve impulse goes in a particular direction, as I'll talk in a little bit, you need to know that these are dendrites. The nerve impulse is actually going from dendrites towards the cell body, whereas the nerve impulse, this motor neuron, is going away from the cell body down the axon. A little bit of a note as well, if you have to label the end here, what we've got here are what we call synaptic end bulbs, or synaptic knobs, or motor end plates, or axon terminals. What you're labeling is the end of the motor neuron, not the synapse itself, so just be wary of that. And again, can you label all of these parts from memory? Because if you're asked this question, then there's some easy marks there, aren't they? So again, I'll just pause and just to give you time, if you wish then to spend time thinking about the labels. Thank you very much. Okay, so here we go. And then there is a diagram there for you to test yourself against. So you can go back and forth with those labels just to test yourself. Okay, so we know there are 3 types of neuron. And you'll notice that the cell body is in different places, and this is how you recognize them. So a sensory neuron, as I've said, takes impulses from the receptor to the central nervous system, and this cell body you will find in that ganglion just outside the central nervous system. In the dorsal root there. The relay neuron, you will see that in the grey matter. You see the cell body in the middle there, and that actually connects at one end with a sensory neuron and at the other end with a motor neuron. So that's connections within the central nervous system. And then finally, the motor neuron, and that actually connects with the relay neuron but takes impulses out to the effectors. So you can see, as I've shown you several times now, that we have a direction of conduction of the nerve impulse. Please, if you're asked a question on that, don't get that wrong. You can see the way they're actually joined up together there, forming the reflex arc. Just one little thing that I did mention, and that is about the dendrites, and they carry the impulses to a cell body. So if you're not certain which bit is the axon and which bit is the dendron or the dendrites, if you're told the direction of transmission, then you can work it out. So the dendrites, as I say, take the impulses towards that cell body. Okay, so it's easy to work things out, and the axons away, as I've shown you in that diagram. Just another little pointer for you. And just some other bits of information as well. Nerve is a bundle of neurons, so there may be lots, well, there will be lots and lots and lots of neurons in a single bundle. And just to tell you about what's happening in the cell body, because that cell body is pretty important, but that's where you'll find the main organelles, so the Golgi apparatus, the nucleolus, the rough endoplasmic reticulum, etc., etc. So don't forget that is where you'll find those organelles. So we'll carry on now and talk about something I mentioned very briefly, which is myelination or myelin. And I always think myelination is just like the production of a Swiss roll, a cake, because you have a cell here. I'm showing you here the cell, and you can see the nucleus in the cell, and this is the cell itself, and the blue part there is the axon. And gradually, as that Schwann cell, as it's called, matures, what happens is it wraps itself gradually around and around and around the axon. You can see there until it's got lots and lots of layers there. The nucleus is still there, but as it's developed, it's actually enclosing that axon in this fatty insulating layer. Okay, so that is going to electrically insulate the axon, but it's not a complete coverage of the axon. It's in just parts. So you get sort of one node, one Schwann cell, and then you get another one next to it, but they're not completely joined together. They're separate cells. So you have these areas which are called nodes of Ranvier, as I've said, where the axons are exposed. And what happens is the impulse jumps from one node between those Schwann cells to the next one. That's called saltatory conduction, and actually it speeds up the transmission of the nerve impulse because nodes are about 1 millimeter apart. Okay, so that is one way of ensuring that you have fast transmission by having these gaps, these nodes of Ranvier, but also the speed of nerve transition is increased also by having larger diameter of axons as well. A couple of pointers for you to remember. So these nodes have what we call special Vogt-gated channels, and that's important in the transmission of the nerve impulse. I'll come to that in a little bit. Now, I just want to contrast those sort of ideas so far with more simple organisms, something like we can see there a hydra, but couldn't resist popping in a jellyfish as well. Many and varied, they're in the same sort of phylum, and actually they're cnidarians, so sponges, corals, sea anemones. And what they've got are just very simple nerve nets. So it's not like us, they don't have a brain and spinal cord. They can't actually detect the source of a stimulus. So they're different from that sort of like our mammalian nervous system. They detect a sort of a limited number of stimuli. Unlike us, they've got a limited number of effectors because of that. So they've got quite a sort of a varied complex sort of system of sort of lots of branching sort of nerve cells, but actually what they do is that they've sort of got lots of branches that will communicate and respond to each other. So they're simpler in that respect. The nerve impulses also travel in more than one direction, and you may recall that in our nervous system, the action potential, the nerve impulse, will definitely just travel in the one direction. They're unmyelinated, so again, different from us. And as I've said, no central nervous system, no main coordinator there, no brain. So, very often a question will come up about sort of comparing the vertebrate nerve system with a simple nerve net system like this, and all the information is there for you. And a quick recap really before we get on to talk about the transmission of the nerve impulse, and that is the two forms of transport. And one is passive transport, which you'll know, simple diffusion, high to low concentrations through the phospholipid bilayer, but also facilitated diffusion, high to low concentrations again, but may involve either a protein channel or a protein carrier. But again, they're passive forms of transport, they don't require ATP. Whereas active transport, as the diagram on the right hand side shows you, it does require ATP. So phosphate is used and ATP is converted into ADP, and what happens is that this is transport against a concentration gradient from low to high, and that is what we're going to talk about next. Because some various experiments have been carried out over the years, and actually famous experiments, there we go, involving frog nerves, found that actually speeds were about 27 meters per second. But we now know that actually the thicker the neuron, the myelinated neurons, carry impulses at about 120 meters per second. I mean, that is fast, isn't it? You know, how were these experiments sort of carried out? Well, what happened was electrodes were inserted into the neurons of frogs, and then what's something called a potential difference was measured using an oscilloscope. So we now know, because of these experiments, that the myelination greatly increases the speed, as I've said, of the impulse. The wider the axon, the faster the impulse, and also we know that the higher temperature, the faster the speed is as well. So just something for you not to forget really about the temperature as well. Moving on to resting potential, I always think resting potential, what a strange name, because it's anything but resting. It's such an active process. And what happens in this part of nerve of sort of the nervous system is that the cell is polarized. So we've got very active processes here. We've got a sodium potassium pump, which is actively transporting sodium ions out of the neuron and potassium ions into the neuron. And actually what we've got, and you might be able to see in this little animation here, is that these pumps are working hard, and for every ATP that's used, they actually pump 3 sodium ions out of the axon, through the axon membrane to the outside, and 2 potassium ions in. And so what that leaves is something that we call a potential difference, where the inside, because more sodium ions have left than potassium ions that are coming in, we actually say there is a difference where the outside of the membrane is now more positively charged than the inside. So as I say, a potential difference exists. So how does a nerve impulse get started? How does it set up in the first place? Well, this happens at the sense organs, and a generator potential is actually set up. So if there is a strong enough stimulus, then what will happen, or maybe weaker stimuli coming from different directions and actually converging there on the receptor, what will happen is receptor cells, which have these gated, what are called gated sodium ion channels, what will happen is that they will open and sodium ions will flood in. Okay, so whereas sodium ions were doing resting potential, were being actively transported out, now they will flood in, and this creates a generator potential. As I say, if the stimulus is large enough, it'll create the generator potential, and then we'll initiate something called an action potential. So this is what, this is what happens when the charge kind of flips. So instead of the inside of the membrane being negatively charged, then actually now it becomes positive. And we say it actually now depolarizes to plus 40 millivolts, because that's the unit from the minus 70 millivolts that we could register on an oscilloscope during resting potential. So this is depolarization. So we need to look at all of the different parts of the action potential. So it has this characteristic shape, and you can see the phases that it goes through. Okay, we sometimes call the undershoot a hyperpolarization, so it just depends which diagram you're looking at there. But the size of that action potential, as long as the stimulus was enough, the size of the action potential will always be the same. So it's independent of the size of the stimulus, and we call that the all or nothing law. As long as the stimulus exceeds a certain threshold value of minus 55 millivolts, then we will get an action potential. So it's always that characteristic shape. So why is it? What's happening all the way along that then? Well, the depolarization, as I say, where the charge has actually flipped, is then quickly followed by repolarization. So let's go through this in the stages then. We've got a threshold here, which in this case has been exceeded. The sodium ion channels first opened, then the potassium, and so sodium then can come into the cell, making it more positively charged. The potassium ion channels then open, and potassium ions begin to leave the cell. And then what happens is we get to this stage where we say it's a refractory period, where the sodium ion channels allow no more sodium ions to sort of enter the cell, and we've got this maximum of plus 40 millivolts. Okay, now repolarization is where the sodium ion channels then will close, the potassium channels will then open, and potassium will flood out to the axon to reduce that potential difference. Sometimes more move out, so we'll have this overshoot, and that's pretty important because that actually stops a new action potential being generated at any point there. and the resting potential is eventually restored using ATP again. And so, as I say, during that period there, an axon can't transmit another action potential. So actually this stops action potentials, going in different directions. They only are able to go in the one direction, an important feature of the action potential there. So let's move on now to synapses. They are very small gaps, about 20 nanometers. They're formed between neurons, but the speed of transmission slows at this point. You can see a very simple diagram there below. So we need to look at how it is that transmission takes place over the synapse and then focus in on how it is that drugs, certain chemicals, will actually affect what's going on there. So we can see here that the action potential arrives, and then calcium channels will open, calcium ions, Ca2+ ions will fuse with vesicles there, which will then travel to the edge, the membrane here of what's called the presynaptic membrane. The transmitter, the neurotransmitter, will actually be released into the synaptic cleft, and it will diffuse across. Hence me saying that the speed there is slowed. The neurotransmitter could be something like acetylcholine, or it can be dopamine. In any case, it will cross the synapse, and then what happens is that it has a receptor site in the postsynaptic membrane. There you can see these blue little receptor sites there, and it'll bind there to those neurotransmitters, and that will trigger a fresh action potential. Now, what normally would happen in this case is that neurotransmitter, if we say it's acetylcholine, it'll normally be broken down by an enzyme, acetylcholine esterase. And so what will happen is it will no longer be bound, it will be taken out of the cleft, it will be regenerated, in the vesicles inside there. But what will happen is that the action potential then will just move on its way, as I've said, and there won't be a merging, there won't be several action potentials merging together. So they're discrete action potentials which will move on from the one neuron to the other. What generally will happen is as I said, the cholinesterase will break down the acetylcholine, and then but other things will stop that merging happen as well, as I've mentioned there. The active transport of calcium ions out of that synaptic knob area there, because then that will stop them fusing with the vesicle and so releasing the neurotransmitter. So several things there can affect this transmission. We need to know about two main groups of neurotransmitters or chemicals that will affect the action at the synapse. One are chemicals like organophosphates. They're insecticides, and they are cholinesterase inhibitors. So what will happen is more acetylcholine will be kept in the cleft, so you get more impulses firing, so that you get more muscle contractions, and that leads to issues. And the other type are the psychoactive drugs that can affect the functioning of the brain in a variety of different ways. But in all of this, you need to think about the fact that these neurotransmitters and the drugs which affect them can be very similar in shape, and that is how they can affect the synapse. So let's look at the organophosphate to start with. So what we've got here is acetylcholine. We've got it being transmitted across the cleft there, and it's binding with the receptor site, and that's what would normally happen. And along would come acetylcholine esterase. There we go, they're shown there by the yellow stars. They will actually then combine with the acetylcholine, they'll break it down, so it'll stop that signaling process. So the impulse will stop firing at that point, and it'll stay as a discrete impulse moving forward. But the organophosphates, what they will do is they will stop that acetylcholine esterase from working. So what happens is, because they stop it from working, the acetylcholine will stay bound to the membrane there, and then you'll get merging of impulses, or you'll get one fired after another, which will cause lots of muscle contractions. So we have a principle, as I've said, that many of these drugs have similar structures to neurotransmitters, and it's a fascinating area of biology, this. And of course, in any exam, as long as you've got your basic principles, you can answer any amount of different questions, as long as you've got those ideas that neurotransmitter can be increased or decreased. You know, proteins needed for the neurotransmitter to be actively transported back into presynaptic neuron, you know, if they are affected by a drug of some sort, if the drug mimics the neurotransmitter, if it blocks the sites, all of these things will affect the onward transmission of the impulse. Okay, so that is how drugs can have sort of excitatory or inhibitory effects, really. Now, what I want to do now is I want to get on to some exam questions. So the first one here says an action potential is illustrated in the following graph. So we can see there it talks about areas A, B, C, D and E, and it's got two areas, X is labelled there as well. And it says what units should be used, on the two axes of the graph shown above. Well, if you're uncertain about how to write the unit out, then make certain you write it in full. And it actually then talks about the membrane potential and the time. So let's look first of all, our units are millivolts or MV. And we can see there that the time there in terms of units there is milliseconds or ms. So if you're uncertain, just write it out in full. Okay, then it says identify the stages of the action potential indicated by A, B, C and D. So A is the resting potential. Remember, anything but resting, it's active, ATP hydrolyzed, and what happens there is that because the sodium ions are being pumped out, then we've got this minus 70 millivolts of resting potential. And then of course along comes a stimulus, there is that flipping of the charge, and B, we have depolarization. Excuse me, and then after that, of course, what happens is we have that period of repolarization where the channels change, and sodium ions and potassium ions, obviously sodium ions no longer moving in, potassium ions then moving, and what happens is that these move out. What happens is that you get this hyperpolarization period there. So those are the labelings A, B, C and D. What is represented by E on the graph above? So I'll just pause there. And actually that is a threshold potential there, because what's happening is that we've got a stimulus which is going to the threshold. Okay, it's not going beyond that at that point to actually generate an action potential. So we might have a maybe some stimuli there that are failed, what I would call failed initiations. Okay, so why is stimulus 2 failing to initiate an action potential? Well, it's because of that all or nothing. It's not quite there. It's not reached minus 55 millivolts, so you won't get that all or nothing response. Let's look at the next question here, because that is basically talking about the same sort of thing here. It talks about the results of an experiment, and you can see we've got some sort of responses going on there, some sort of initiations, but they're failed initiations. The threshold has not been reached there, and that's, remember what I said, it's all or nothing. And then it says explain the opening and closing of the ion channels, resulting in depolarization and repolarization. So what we've got here, remember, for depolarization, and again I'll pause there a little bit, sorry, so that you can see that, remember, sodium channels are opening, sodium ions are diffusing rapidly in, and then the inside becomes more positive relative to the outside. So we've got that flip-in charge from the resting potential, 40 millivolts, as I say, always the same size. And repolarization there, I think I went over it a bit too quickly earlier on. So we've got the sodium channels closing and the potassium opening then as we go down that action potential slope. We've got potassium ions diffusing rapidly out, and then we've got that negative relative to the outside, so gradually the resting potential will return, but obviously we'll get a maybe hyperpolarization first. So we know that all of these values are potential difference, remember? Okay, another common question then for us to look at there, and this is one where you're shown an image. I mean, I've spoken before about images. You know, as a biologist, you need to be able to label things and explain what, in this case, the electron micrograph is showing. So it's asking us about the name of a substance, and the second one, the name of a cell. Right, so I'll pause there while you think substance, cell. Okay. So the substance is our myelin, our fatty sheath, which is Swiss rolling around the axon. Okay, and the name of the cell is actually our Schwann cell. Okay, so don't get those confused, because it would be easy to think, oh yes, they're asking me, they're pointing to the Schwann cell. They're not, they're asking you about the substance. So again, a good exercise in reading the question carefully. And then it says, explain how the substance secreted by these cells and their arrangement along the neuron affects the speed. So we know from what we've said before about how it is that myelin is going to affect this, it has a sort of an insulatory effect, but it's the nodes of Ranvier. Yes, so I'll just pause while you think about your answer there, because that is 4 marks. There's a lot of detail in there. So now I'll reveal then that it's going to electrically insulate those parts of the axon. It's going to prevent depolarization other than at the nodes. Okay, those nodes of Ranvier, and it's only at this point that actually you get the depolarization. So it'll jump from 1 millimeter to the next node and so on. And that is how nerve transmission is incredibly fast. Again, a nerve diagram to think about labeling. I just want to sort of point out that the nerve transmission is going to be from dendrites to cell body and down the axon. So you might want to pause now, or I will pause very briefly while you think about what are the 2 types of effectors. Where in the spinal cord would you find structure X? So that's the cell body at the top there. And in which spinal cord root would you find structure Y? So have a little think about those 2. So I'm just going to reveal. So A, we've got dendrites. It's pointing to 1, so maybe you might have put dendrons, so dendrites, but also dendron is acceptable there. For B, it's a little bit unclear. Is it pointing to the axon or inside to the axoplasm? So, you know, any of those would be acceptable in this case. C, there are good old nodes. So there are nodes of Ranvier. And then at the end, as I've said before, don't put synapse. It's a synaptic knob, the motor end plate, an axon terminal, but it is not a synapse. It's a structure of a motor neuron, and a synapse is not the structure of that. And then finally, I think the 2 types of effectors, the muscle and the gland, and they're not asking for specific names, so just remember it's types they're saying there, muscle or gland, and then the last 2, X you'd find in the grey matter because it's cell body, hence giving its colour, and you'd find because this is a motor neuron, you'd find that axon in the ventral root at the bottom coming out. And just another question. So again, identify X and Y on the diagram. So again, nice little bit of, nice couple of marks if you can get it there on why they're so called, and L and Z. I think I've probably been through all of that. So I'm just going to pause, but then I'm going to move quickly on. So, okay, if you've had a chance to think now, you've sorted your dorsal and ventral, you've sorted your white and grey, obviously the cell bodies as opposed to not, and L and Z, the dorsal and the central canal, which I mentioned earlier. And again, these questions, you'll find a range of them which are focusing on lipid and myelin. So just try them for yourself when you've got more time. And then just the last question I'm going to go through, which is lovely Hydra and the nerve net. The question says, draw in a nerve net. And I've tried my best there to show you how I might draw it in. And then it asks you to show 2 ways in which it's different from the nerve system of a vertebrate. And remember, no source, no CNS, no brain. The cells are unmyelinated, so it's slower transmission. The action potentials go in more than one direction. Small number of effectors, so the number of stimuli it responds to is small as well. And then this last question is one for you to try here on organophosphates. So again, I will pause and you have a look at this at your own pace, but it's all the things I talked about earlier on. So just have a look at the key parts of the question there, and then obviously then as I'm revealing this, you can sort of work through this at your own pace. Particularly there, you know, a nice little question, but remember the question is about how you would decrease, how the psychoactive drugs would decrease the rate of synaptic transmission. So please think about that. Okay, I'm going to leave you with the dice sheets and with the answers to the dice sheets for this topic. And all you need for that, I just feel the need to say a little bit more about this today. All you need is 1 die or maybe even 2 dice, and you can use it independently where you can actually throw the dice yourself, and then you just think, right, I'm going 4 across and 6 down or whatever, and test yourself. Or you can actually work with somebody else and they can generate some random questions which they can fire at you, no pun intended, with nervous system, in order to use it as part of your revision. So there's, you know, an empty one as usual. There's the answers for you to try yourself, and it just remains for me really to say thank you very much for sticking with me.